There you go.
Thanks. Thanks, everyone, for coming this morning. It's really a pleasure and an honor to be here today. And boy, the room is already full. Everyone's been on time. I do want to recognize the other speakers for a minute, because I think you're in for a real treat today. After I speak, Joseph Buttigieg will speak about a therapy he's invented that has potential to help people with MS. And then Robert Lepere will also speak. He's an expert in marijuana, and with all the hype with marijuana, he'll be able to help us sort out the real from the fluff. And in a similar way, James Marriott is actually heading up a study in one of the stem cell therapies. So again, there's been a lot of news about that, and he'll be able to educate us in detail about that, and again, separating out what's real from maybe what is not so real. Okay. There we go. You're going to see this slide a lot. It's just incredible. My team's in the back here. I asked people to come and give up their Saturday and volunteer, and we had 17 people say yes, and I just can't thank them enough, and I'm going to ask you to give them a round of applause right from the beginning. This is part of that team. This is the team that's actually doing the work at the bench and the research that we're doing as well as taking care of all the clinical research tools we need to get clinical research out to people. And every one of these people is here today, and you'll meet them later on. Okay, so we are the Office of the Saskatchewan Multiple Sclerosis Clinical Research Chair, and we've been open for about 18 months. And so who are we? You've met my team already. Where are we? Fortunately, we're all in the Saskatoon City Hospital. Not only my offices are there, but the Chemical Neuroscience Research Center is there, the MS Clinic is there, the Department of Physiotherapy is there, and the MS Drugs program is there. So it might be the only one in Canada that has everything under one roof, and this really allows for great collaboration and communication. What do we do? I'm going to show you today that we collaborate and create partnerships. I'm going to give you very specific examples of that. Our research is into the cause of MS. We're interested in treatments and cure for MS, and we give compassionate and we think cutting-edge care now. Right here in Saskatchewan, you don't need to leave the province. So the 4 C's. What is our philosophy? So, we really think people, including people in this room with MS, will help us cure MS. Why do we do MS research? Everyone needs resources to do research. There's a lot going on medically in the province, but here we think MS touches just about everyone in Saskatchewan. Well-funded research is permanent. It creates jobs. It attracts talent and keeps talent here, and we have evidence of that as well. It inhibits complacency. Researchers, I think, are forever curious, and we never lose that curiosity. And it provides the best clinical care in Saskatchewan. There's no doubt that the backbone to great clinical care is research, and that's been shown around the world, and now we're beginning to set that up here. So don't leave the province. You can come right here. So how common is MS in Saskatchewan? Worldwide, the rate of MS, the prevalence of MS, is about 1 in 3,300. And in the States, it's about 1 in 1,000. And the difference between working here and the United States is I felt like everyone knew of someone with MS. Well, here in Saskatchewan, everyone knows someone with MS, and I was kind of corrected. So again, in Canada, the rate is about 1 in 500 to about 1 in 300. And it seems like the highest rate actually is here in Saskatchewan. And so I used to say everyone knows someone with MS, but I was corrected by our health minister, Jim Ryder, who said, no, actually, Michael, everyone knows 2 or 3 or 4 people with MS, actually. And he reeled off 3 or 4 or 5 names right off the bat, including 2 family members. So what is MS? It's an inflammatory disease. White blood cells, for reasons we don't understand, are attracted to the brain and the spinal cord. And actually, myelin, the coating of neurons, begins to be destroyed. I'll show you a photograph of that. And in an area called in the brain called an MS plaque, and it's a different kind of plaque than anywhere else in the body. In medical terms, we use the word plaque a lot, but this is a unique kind of structure in the brain and spinal cord, and it results in either relapses or worsening or progression of disease. So there are different types of MS, and in some people, MS gets worse, and we'll talk about that as it relates to our research. But if we look at the top part of the graph, and on the y-axis is disability, and on the x-axis is time, some people are fortunate enough to have benign MS. They have a few relapses through their life. They recover completely, and that's benign MS. That's usually the exception to the rule. Most people start with relapsing remitting MS, and we can see here, I can't point to all of them, but again, someone's going along and they have their first attack, maybe an optic neuritis or spinal cord problem, and then they recover and they continue to have attacks over time. Some people with relapsing remitting MS will switch to what we call secondary progressive MS. It happens secondarily to relapsing remitting, and these people actually get a little bit worse without having attacks. And when that happens, we think that's related to nerve cell damage, or what we call neurodegeneration. And people with primary progressive MS never have attacks, and they slowly get worse over time, and we think, again, this is related to nerve cell damage. All research starts with the patient, and I'm going to show you that today. We think about people with MS all the time in the lab. There we go. Here's an MRI scan of the brain. So who's had an MRI scan? I have for other reasons, by the way, but you know, when I started this 20 years ago, there was still a significant proportion of people with MS who hadn't had an MRI scan yet, and I'm glad to see that that's been corrected. The yellow circle is really an MS plaque. It's incredible we can see it in live people. On this particular scan, it's bright white, it's ovoid, it's greater than 3 millimeters in size, and it's near the middle of the brain. So this is, when we talk about an MS plaque, this is just a classic one. This is what we're talking about. And let's look under the microscope now. So again, to your right is that same plaque, but now we're going to look at it under the microscope. Okay? And what we first see is shown in the middle of the slide are a lot of black dots, and that's actually a blood vessel filled with inflammation, with white blood cells. And if you look closely, those white blood cells, again shown by the black dots, are starting to enter the surrounding brain. Early in disease, we know there's loss of myelin. And so in the bottom of the left, the blue staining is normal myelin, and you can see in the upper right-hand corner of that same slide a large area of white, and that's demyelination, or loss of myelin in the brain in those same areas where we find the white blood cells and the inflammation. At the same time, the myelin coats nerve cells. And we actually thought in the beginning this was only a demyelinating disease, and nerve cells died secondarily. Now we know both are under attack at the same time. And if you look to the bottom right, and you just follow kind of the yellow lines through all that red, so you see the green circle, and you follow that yellow line should be perfectly smooth. And it's not. You see it's starting to be eaten away, and that shows that that nerve cell is being damaged or undergoing neurodegeneration. Okay, and so now this is kind of the research part of my talk. We're interested in stress granules because we think a protein named A1 is attacked. And just to orient you, this is what a cartoon of a nerve cell, and the large red area in the middle is the nucleus, and the surrounding area is the cytoplasm. And the red area is this protein called A1, which is also on the upper right-hand side of the slide. And what I'm going to show you today is people with MS make antibodies to A1. Okay, a certain percentage make antibodies to this protein. And in that setting, there's inflammation. Okay, and we're going to show you what cytokines are. And inflammation are those substances secreted by those white blood cells that have entered the brain or spinal cord. They're there secreting abnormal substances, and they're damaging the brain. And even some people have DNA mutations, or have mutations or abnormalities in their DNA that also affect A1. So all of these all these changes in the immune system, we think, at least in our work, is focused on a protein called A1. What happens is when, and we'll go back just to make sure everyone caught that, so if you make antibodies or cytokines have DNA mutations, A1 gets lost. It goes from the nucleus to the cytoplasm as shown there. Over time, it gets packaged. It gets packaged into stress granules, which we think are bad for neurons, and that's shown there. And we think this triggers nerve cell damage or neurodegeneration. Okay? And I know my time is limited, but would anyone like to see that again? One more time? Well, yeah, I figured. Let's go through it again. Well, let's go through it backwards. Maybe that helps. So we'll go backwards, but we'll go through the whole story again because it is a complicated one. So A1 is one of the proteins in a nerve cell. It lives in the nucleus normally. MS patients make antibodies to it. That means something's going to happen to that protein. It's not going to work properly. At the same time, white blood cells have entered the brain secreting all sorts of abnormal substances related to inflammation, known as cytokines. One of them is known as interferon gamma. We'll look at that data today. That also affects how A1 works. Some people have mutations in their DNA, and all of these can cause A1 to dysfunction. And again, it lives in the nucleus. When any of these things happens, it gets lost, it gets stuck in the cytoplasm, where it becomes packaged into stress granules. And we think when this happens, the stress granules, which usually protect nerve cells, begin to damage them, and that triggers nerve cell damage. So let's look at some of the science. Okay. What research are we doing? To step back a little bit, in 18 months, we've submitted 25 grants or contracts, both out of our office and people we collaborate with, worth more than $5 million, and we're inching our way up into the most important category, which is the awarded category. So let's look at some nerve cells. Okay, and I'm sorry I can't get to all of them, but I think I can walk you through it. This is a high-powered view of human brain, in both a normal person and a person with MS. Our friend A1 is mostly all in the nucleus. There's a little bit in the cytoplasm, but this brown staining is A1 all in the nucleus in a normal patient. And you can see in MS, this is the nucleus here. But that A1, like we showed, gets stuck in the cytoplasm and actually forms granules, which I think the easiest way to visualize are like granules of sand or grains of sand. This marker, TIA1, so now we know it's stuck in the cytoplasm. It looks like it's in a granule. There are a lot of granules in the brain. Is it a stress granule? And this marker, TIA1, is specific for stress granules. It too gets lost from the nucleus, but it actually, the nerve cells form stress granules as well. So A1's in the cytoplasm where it normally is not, and so is TIA1. And then using a more sophisticated technique where we can actually double label the cells and actually ask, yeah, we see granules here. We see stress granules here. In science, we have to prove they're one and the same. And again, A1 is green here. Here's our nucleus. And the granule is here, right here, A1, same staining as here. And TIA1, same staining as here. And when you merge them together, green and red make yellow, and you can see that A1 is stuck in stress granules, which we think are bad for the brain. I do want to recognize Hannah Salapa, who's a graduate student in my lab, and she's done most of this work. So again, we've talked about that antibodies. Antibodies can cause stress granules. How do we know that? This is what a nerve cell looks like under the microscope in a petri dish. Nucleus is blue and the cytoplasm is green. We add normal antibodies and the green stays, but you can see when we add antibodies from an MS patient, stress granules begin to form, these green blobs. Same thing, this time in those same neurons, we added cytokines, those proteins that are secreted by the inflammatory response. Again, we'll start in the middle. A1, which usually lives in the nucleus, now it gets stuck in the cytoplasm. It forms stress granules, and we've actually showed that this protein, again, A1, is in that stress granule right here. And finally, third, people with mutations to A1 is also, again, abnormal. So here, using special molecular techniques, again, looking at nerve cells under the microscope, nucleus, cytoplasm. You put normal A1 into these cells, and you see it fills the nucleus really quite nicely, as shown here. But you put abnormal or mutated DNA into those same cells, and stress granules form in the cytoplasm, as shown here. So what can we do about it? One, fortunately, we think stress granules are now bad for the brain. There are already drugs out there, experimental drugs, that actually can dissolve stress granules. This is not our work. This is work from another scientist. And you can see, again, if you put, you can see cells with stress granules, as shown here, nucleus, and then all the green dots are stress granules, and you add this drug in culture, and you can see some of those stress granules disappear. So we're actually testing some of these drugs in the lab. So why is this important? Always a critical question, something we have to answer to scientists and colleagues and peers, but something we have to answer to all of you every day. right? Why would this matter? We think that these stress granules and abnormal formation of A1 in the cytoplasm and the combining of those two actually trigger or are part of neurodegeneration. We think it's part of why people with MS get worse, whether they're on medicine or not, and our plan is to try to stop that and level out these lines. So let's talk about kind of more research in the province. We also see patients with MS in Saskatoon City Hospital. And I just want to let you know that we see more every year, and we appreciate when you come to the clinic. And most of our staff is here today. And of course, you all come from all parts of the province to come see us, and we appreciate that. Okay, we talked about the 4 C's. You know, we've talked about looking into the cause and cure for MS, right? The other thing we do is really try to create collaborations, and these are things that I really cherish. And again, we're looking for partnerships that help MS research here in Saskatchewan. Okay, we want to bring research here. We want to be competitive here. We want to be a world-class MS Center of Excellence here, right in Saskatchewan. And these are some of the people. I have to admit, I have to put my wife first because, you know, after 28 years of marriage, we know who goes first anyway. And she's here today. And we'll talk about that a little bit later as well. But these are all of the people who have begun to help us at the University of Saskatchewan. And again, these are really true partnerships where both sides are completely involved and get credit for their work. I do want to talk about 2 special partnerships we have at the University of Regina with Joseph Buttigieg and Mohan Babu. So Dr. Babu came to us. He's got some cutting-edge technology that is going to look at mitochondria and other causes of MS, and we've started that collaboration as well. And as Joseph will talk about later, he's invented a medication that he thinks will help MS. And again, we're here to partner and promote and improve MS throughout the region. And Joseph's student actually came into our lab for a couple weeks to learn those techniques and bring them back to the University of Regina. So those are really the kind of beneficial partnerships that we're looking for. They're not only regional, but they're actually national. Same thing here. These are people that are really helping our work here in Saskatchewan and helping put Saskatchewan on the MS map. And of course, internationally as well, some from my old scientific friends in the States. Again, the idea of creating strong and lasting and mutually beneficial partnerships. So again, I think I'm just about done. I do want to recognize my team here again. They've been fabulous. Am I somewhat on time? Right on time? Fantastic. I tried to do it that way. And again, I just want to thank everyone to help make this possible, including my family. We were fortunate enough to be scuba diving at the Great Barrier Reef just last summer, which is a lifelong dream. Saskatoon and Saskatchewan was named as one of the 52 places to visit in 2018 by the New York Times. And you know when the New York Times says it? And here we are in the middle. And again, thanks to the MS Society and everyone who's created this MS chair and really has helped put us on the map for MS research. Thank you.
Thank you so much, Dr. Levin. And I know we're actually going to hear more from you throughout the day. So our next speaker is Dr. Joseph Buttigieg, and he conducts his research at the University of Regina, as just mentioned, and was recently featured in the news media, which has generated a lot of interest.